Hello everyone. So this is the first chapter of the second section of the course. We are continuing to examine the cellular mechanisms that contribute to learning and memory. And in this chapter, I'm going to introduce the idea that LTP is not a singular phenomenon that onsets when stimulation occurs and simply lays down a pattern of enhanced synaptic communication. Rather, there are distinct waves of activity in LTP that can be thought of as stages or phases, where a cascade of mechanisms are employed. These stages include an initial stage wherein kinase-mediated alterations in existing proteins bring about reversible enhancements in the postsynaptic response to presynaptic stimulation. There's a second stage in which new proteins may be synthesized from pre-existing RNA. And there's a third stage that requires signaling through to the genome. The first stage of LTP encoding is known as short-term potentiation, and it lasts for about 15 to 30 minutes. The second stage is part of the late stage LTP, and it lasts for about 2 to 3 hours. And the third stage is fully late stage LTP, and it lasts 6 to 8 hours in the hippocampal slice, being limited really by how long that ex vivo tissue remains functional. Hypothetically, late LTP can last from weeks to a lifetime in a healthy intact animal. We can explore these stages and some of the underlying mechanisms using three models of LTP induction known as LTP1, LTP2, and LTP3. I have to pause and remark about what an imaginative set of names that is, LTP1, 2, and 3. Some poor unrecognized scientist really was expressing his creativity with those names. Not. Well, anyway, I modified one of the diagrams from your textbook to depict the fundamentals of LTP1, LTP2, and LTP3, and to illustrate the principles that we will talk about in this chapter. The early stage of LTP consists of the post-translational modifications of proteins that I spent much of the first 4 chapters talking about. The second stage includes new protein synthesis, but does not require genomic signaling, and the third stage requires genomic signaling. And of course, these mechanisms of LTP all have implications for our understanding of learning. There are many lines of evidence that these same mechanisms underlie various kinds of learning in intact animals and even in humans. And we'll delve into all those data in later chapters. But for now, let's examine these stages. An interesting series of studies depicted on this slide reveal once again that LTP is not necessarily stable after it is first induced. But in contrast to the previous preparations we saw, where LTP was disrupted by addition of depotentiating low-frequency stimulation, in this case, there was actually no disrupting influence. There was just a difference in the stimulation parameters. Abraham and collaborators from Mike Dragoneau's lab stimulated some hippocampal preparations with standard strong tetanic stimulation. And you can see the robust LTP represented by the closed circles in the graph at the upper right of the slide. But then they stimulated additional preparations with tetanic stimulation, but in this case, they used stimulation that was not quite as intense. It's labeled on the graph as weak stimulation. But I don't want you to confuse it with weak low-frequency stimulation that's used to induce long-term depression, or LTD. This is actually weak tetanic stimulation, or maybe it should be something like tetanic stimulation that's not quite as strong as the tetanic stimulation that induced long-lasting LTP in the upper curve, but rather slightly less strong, so it only induced a transient LTP that diminished rapidly and is represented by the lower curve on the graph. But you can see how that lengthy but, well, more accurate description wouldn't quite fit on the graph, so instead they labeled it weak. In any case, you can see on the upper graph that they were able to induce a form of LTP that looked almost as strong initially, but then faded away over the course of a few hours rather than continuing for the duration of the viability of the preparation. And the lower graph on the slide extends the concept of LTP beyond the constraints of the duration of viability in an ex vivo section to LTP in the hippocampi of living animals. This lower graph shows LTP in a strongly stimulated hippocampus that was followed for a full year and was still going strong at the end of that year. So we can see that potentiation can be a short-lived phenomenon that lasts only a few hours or less in the weakly stimulated hippocampal slice, to LTP that seems permanent, or at least nearly permanent, after strong tetanic stimulation in an intact hippocampus of a living animal. So this brings up a question. How can synaptic potentiation endure over long periods of time in the face of molecular turnover? The modifications I described in the first 4 chapters all consisted of reversible changes. You can phosphorylate a receptor, for example, and that will cause it to be inserted into the membrane. It could even be stabilized there. But we know that phosphorylation is a reversible action. So why doesn't the synapse just return to its naive state after a while? Certainly it did in the case of the weak stimulation in the top graph, and certainly there should be a tendency to slip back to the naive state if all that was supporting the potentiation was the presence of some phosphorylated proteins. And let's just assume that the protein phosphorylation somehow became irreversible, so they never lost their phosphate group. Now that does not happen, but I'm kind of making up a fairy tale hippocampus here in which phosphorylation is a permanent change. It still wouldn't be sufficient to maintain a potentiated hippocampus. You see, all proteins have a half-life. They are eventually tagged and transported off to the lysosome to be degraded. If the receptors and other phosphorylated proteins were just taken off to the lysosome, the potentiated signal would be lost. And even if those proteins were replaced by newly synthesized proteins, the newly synthesized proteins would not be phosphorylated. So the synapse would not be potentiated just on the basis of eventual turnover and replacement of proteins. So you see, phosphorylation of proteins in the spine by protein kinase A, protein kinase B, protein kinase C cannot be the whole answer. These mechanisms are very efficient for initial induction of LTP, but something more must be going on in order to continue the potentiation of the synapse beyond the lifespan of the phosphorylation events, or even beyond the lifespan of the proteins themselves. So, what is the answer? Any guesses? Oh yeah, well, that one's on the slide too. So you can see that the answer is increases in gene expression and protein synthesis. And of course, the protein synthesis would have to include proteins that would support restructuring and ongoing potentiation of the synapse. Now, there is certainly evidence for this, as strong tetanic stimulation produces increases in hippocampal RNA, and memory is associated with increases in protein synthesis. However, a cascade of signaling that goes from synaptic mechanisms of phosphorylation to genomic signaling to activate transcription of RNA and translation into proteins still cannot account adequately for the continuing presence of LTP in the first hours post-potentiation. Observe the rapid decline in field potential responses to Schaefer collateral stimulation in the weakly stimulated synapse in the upper graph. And then consider that activation of a genomic cascade of transcription to new protein after an inducing stimulation like LTP takes at least a few hours. The decline in the signal strength in the weakly stimulated synapses in the upper graph appears to reflect the decay of the kinase-mediated signal. So if we had to wait for a genomic outcome, the potentiation would have to partially collapse and then recover. But it doesn't. Look at the upper set of points in that upper graph. They don't decline at all. So I'm going to propose a mechanism that looks something like the mechanism I'm showing on the next slide. The figure at the bottom of this slide shows a drawing I made to illustrate the three hypothetical stages. I haven't attached any specific time frame to it, but they do appear to occur in this sequential order. The first stage depends upon post-translational modifications, the mechanisms we've discussed so extensively across the first 4 chapters, and that are depicted as a reminder in the graphic at the upper right. The second stage is dependent upon new protein synthesis, but not on genomic signaling. And the third stage is dependent upon protein synthesis with genomic signaling. I'm going to explain that. If we apply strong, high-frequency stimulation to a hippocampal section, of course we would get LTP that should last for hours. Well, of course. And this is shown in the graph on this slide, where you can see that the control cells show enhanced EPSP amplitude recorded with an intracellular electrode. Now, what do you think would happen if we interfered with calcium influx? Yeah, of course, it is also shown on the graph. If you administer BAPTA into the postsynaptic CA1 pyramidal neuron, which you can do by micro-injecting it through the glass recording electrode, shown in the upper right, you can completely block the induction of LTP. I think I actually mentioned BAPTA before, but in case I didn't, BAPTA is a calcium chelator. It binds up calcium. So imagine if you inject it directly into the postsynaptic neuron and then apply tetanic stimulation. The NMDA receptors and voltage-gated calcium channels can open, and the metabotropic signaling can cause efflux of calcium from the endoplasmic reticulum stores. However, as soon as the calcium enters the intracellular fluid of the spine, it will be bound up by BAPTA and will not be available to activate calcium-sensitive proteins that we know are essential for induction of LTP. Similarly, if we inject PD-98059, a drug that blocks kinases, then the induction of LTP by tetanic stimulation is also a big fail. So these experiments remind us that calcium and kinase activity are essential components of LTP induction, and that these signals and a subset of the targets they activate, like AMPA receptors, actually constitute the first phase of LTP. Based upon everything we learned in chapters 1 to 4, we know that interfering with them will prevent LTP induction. Now let's look at some additional manipulations. In the graphs on this slide, anisomycin or actinomycin were injected into the hippocampus, and the effects on hippocampal LTP were examined. So let's talk about anisomycin and actinomycin for a minute. You might recognize the names of these drugs because they're well-known antibiotics. So you might be asking, what do antibiotics have to do with LTP? Or what do antibiotics have to do with learning and memory? Well, to answer those questions, we need to examine the mechanism of antibiotic actions. When you have a bacterial infection, bacteria multiply at a very rapid rate. Under ideal conditions like moist, air-exposed, roughly 37 degrees Celsius conditions in your sinuses, or in a break in your skin, or, well, especially in an idealized incubator in a laboratory, well, bacteria will double their population about every 20 minutes. So, how do antibiotics fight that? They disrupt protein synthesis in bacteria. The multiplying bacteria need to engage in a lot of protein synthesis to form new bacteria. If the antibiotic can diminish that rate of reproduction, it may buy some time for your immune system to get a handle on the infection and fight back by eating up the bacteria. So the antibiotics are protein synthesis inhibitors, and they can actually be used in experimental preparations to see if biological functions rely on protein synthesis. Now, the graphs on this page actually summarize many experiments over the years, including experiments conducted by Krug and collaborators in 1984 and 1988, and by Huang and Kandel in 1994, and they were reported by Kelleher and colleagues in the journal Neuron in 2004. In the upper graph, you can see that these experimenters administered a single train of 100 Hz stimulation to Schaefer collateral inputs to CA1 pyramidal cells and found decremental LTP. I mean, there was an initial induction of LTP, and it decayed over the next 1 to 2 hours. The control animals that got the single train of stimulation are shown in the black line on the graph. And you can see that the pattern of response is similar to the pattern in the experiment by Abraham and collaborators in the second slide of this lecture. Now, I want you to notice that some of the preparations were treated with the protein synthesis inhibiting antibiotics, actinomycin, depicted in red, or anisomycin, depicted in blue. The lines for both of these treatments lay right on top of the line for the untreated controls. So it's clear that protein synthesis inhibition does not affect the early stage of LTP. In other words, the early, labile, and degrading stage of LTP is not dependent upon protein synthesis. And you might not be surprised about that, because we've already in chapters 1 to 4 discussed so much about the kinase activities that are responsible for the early part of LTP, for the induction of LTP. However, let's look at the bottom graph. In these experiments, the sections were treated with 4 trains of tetanic stimulation, a paradigm that's sufficient to induce long-lasting LTP in hippocampal synapses. In these experiments, both antibiotics disrupted late LTP. Let me be really clear about this. If you look at the graph, you can see that the early induction phase of LTP is intact. The protein synthesis inhibitors did not interrupt the first stage of LTP, just as in the upper graph. However, Then the potentiation was lost in comparison with the enduring potentiation that was seen in the stimulated sections that were not treated with antibiotics. But I also need you to pay attention to another important point. Do you notice that the effect of anisomycin, depicted in red, is faster than the effect of actinomycin, depicted in blue? There's a very fundamental reason for this. Well, all antibiotics are not created equal. Some antibiotics are protein synthesis inhibitors that specifically block translation from RNA to protein. Anisomycin is one of them. Other translational inhibitors include emmetine, cyclohexamide, and lots of others. But then there are other antibiotics that are protein synthesis inhibitors that actually work at the level of transcription rather than translation, blocking the transcription of DNA to RNA. This includes actinomycin. Let me repeat that. Anisomycin blocks translation of RNA to protein, and actinomycin blocks transcription of DNA to RNA. This is actually illustrated in the diagram on the lower left. So in the graph on the lower right, we can see that anisomycin disrupts LTP earlier than actinomycin does. And it does so because it interferes with protein synthesis that is not dependent upon genomic action. In other words, it interferes with translation of RNA that is already present. The later effect of actinomycin is due to disruption of genomic actions. In summary, the first stage of LTP depends upon post-translational processing of proteins that already exist in the synapse. The second stage of LTP depends on translation of RNAs that already exist. And I'll show you how this can be very readily available for the synapse. The third stage of LTP is dependent on a signal that must be conveyed to the nucleus and will then activate translation of specific sets of genes so they can provide additional new proteins to support the potentiation of the synapse. We have three broad waves of molecular changes in the cell, each of which plays an essential and sequential role in the organization of synaptic potentiation. And one more important point is that Huang and colleagues in 1996 showed that these protein synthesis independent and protein synthesis dependent properties of LTP are seen in all three major intrinsic pathways in the hippocampus. The perforant path inputs to the dentate gyrus granule cells, the mossy fiber inputs to the CA3 pyramidal cells, and the Schaefer collateral inputs to the CA1 pyramidal cells. So let's continue to explore these waves of biosynthetic activity that underlie your abilities to learn and encode memory storage in your brain. The cartoon on this slide is meant to portray that each spine is equipped with the machinery that can process local protein synthesis from mRNA at the base of the spine. It may not be the best representation because it's really just a silly cartoon and it requires some explanation. But other diagrams that are available on the internet get hung up on details of the mechanisms, so they're complex and don't clearly represent the point that I want to make on this slide. This simple cartoon actually does. What it shows is that at the base of each spine, it shows many ribosomes. Consisting of the large 50S subunit and the small 30S subunit, or the large purple blob and the small purple blob, right? You can see those. The two spines on the right side of the dendrite are inactive, and so the polyribosomes are inactive. However, the spine at the left side of the diagram depicts a stimulated input in which the spine is engaged in activation of kinases and other signaling molecules, and the polyribosomes are actively engaged in translation of mRNA right there at the base of the spine, or even translocated to the head of the spine to synthesize new proteins from RNA that is constitutively available and activated or translated in an activity-dependent manner. The mRNAs that are available and are specifically translated by the activated polyribosomes include CAM kinase 2, AMPA receptors, and something called activity-regulated cytoskeleton-associated protein, or ARC. We've obviously seen CAM kinase 2 and AMPA receptors, so it's not too surprising that they are among the species of RNA that is readily available to be involved in potentiation of active synapses. But ARC is a new one, and I'm going to show you more about its function in some really exciting experiments that I'll describe in the next chapter. Anyway, the result of all this activity is that these constitutively available mRNAs are translated into essential proteins directly in the activated or potentiated spines long before any genomic cascades could begin to provide support to the potentiated synapses. For the next experiment that I want to show you, I first need to remind you that the Schaefer collaterals innervate both the stratum radiatum, or the dendritic field where the apical dendrites align, shown here and here, and the stratum oriens where the basal dendrites align, shown here and here. We saw that previously in the experiment where heterosynaptic LTD was first discovered. Remember that? Stimulation of the Schaefer collateral inputs to the stratum radiatum dendritic field produced depression, heterosynaptic depression, of the responses of the dendritic field in stratum oriens. So anyway, we can actually target the stimulation so that one dendritic field or the other can be selectively potentiated by the Schaefer collateral inputs. Bradshaw and colleagues in 2004 found that local application of the protein synthesis inhibitor emetine in the stratum radiatum disrupted the ongoing expression of LTP for Schaefer collateral stimulation targeted at the apical dendritic field in the stratum radiatum, without interfering with stimulation-induced potentiation of the basilar dendritic field when Schaefer collaterals targeting the stratum oriens were stimulated. These effects are illustrated in the two graphs on the left side of the figure. The lower left graph shows LTP in the stratum radiatum when stimulation was administered to the Schaefer collateral inputs to the stratum radiatum. The blue line shows ongoing LTP in the control slices that were given tetanic stimulation and no emetine. The red line shows the decaying LTP after just 2 to 3 hours in slices that were stimulated in the presence of emetine administered to the stratum radiatum. The